So thank you for the introduction and for the possibility to give some insights into how, let's say, we perform large-scale cohort studies and multi-omics, and in particular also metabolomics analysis. So I'm coming from the field of cardiovascular research, and currently the risk assessment for cardiovascular disease is based on the well-known classical risk factors. So, for instance, smoking, age, sex, the lipids, body mass index, and mainly also on classification by clinical patterns. But it does not comprise any molecular or genetic factors that might reflect the heterogeneity of cardiovascular diseases. And for that, so my group is trying to integrate data from different resources. We also try to use novel methods, multi-level approaches, and my team is an interdisciplinary and translational team. So I always call it our translational circle. So the resources that we have at the workflow we are trying always to approach is that together with our clinical partners, we generated huge cohort studies as well as clinical trials. And this is based on population cohorts, on disease cohorts, so with disease subjects, and clinical trials conducted. And from each of these subjects, we are trying to get biomaterial and have set up a large biobank. And then we go different routes. So on one hand, we use the biomaterial to conduct omics analysis. Here then also statistics and bioinformatics plays a role. And based on the results from those omics analysis, we are then following candidate molecules into molecular and mechanistic insights. So this is, let's say, basic research. And on one hand, based on the omics analysis, but also on our molecular evidence, we are then trying to go back to the cohorts and try to find circulating biomarkers that either reflect what we found in the molecular analysis or are, let's say, a follow-up on our results. And by that, we can always try to start with clinical data, with the clinical research questions, and go the way, on one hand, forward to molecular analysis, and then try to translate it back. And the resources we are having on a population or on a cohort level is, on one hand, the population-based cohorts. Here in Hamburg, we have set up the Hamburg City Health Study, HCHS, which is currently still recruiting. So 20,000 subjects have already been recruited, and it shall be 20,000 more subjects. What has already been introduced is our EU projects of population-based cohorts, large consortium, which is called BiomarCaRE, and this will be detailed in a few minutes, where we have data and also biomaterials from over 300,000 subjects. We also set it up a disease-based cohorts, so all patients that come to the clinic. This is called our clinical cohort studies. And as I said, also the individual clinical trials. And from each of the individual cohorts and subjects, we have a data bank of the clinical phenotype data, lifestyle, endpoints, because most of them have already been followed up. And we can perform cross-section and longitudinal analysis. We have a large biobank. And with these data, then we go on one hand, also depending on the research questions for the omics analysis. And as I said, this translational circle we have tried also to implement in the BiomarCaRE project. So I try to explain briefly what BiomarCaRE is and why we have initiated this, and then I need to go back way in the past to the WHO Multinational Monitoring of Trends and Determinants in Cardiovascular Disease project. It's maybe most of you might know it from the MONICA project. And it was in the former times that there was a rise in chronic diseases, in particular cardiovascular disease. It was, although much known about this, but at the population level, it was a problem. And that's why the researchers established the MONiCa cohorts across Europe. And based on these MONiCa cohorts, then in the EU FP7 project, the MORGAM project, MONiCa Risk, Genetics, Archiving and Monograph was established. And here a lot of population-based cohort studies across Europe were on the data level collected, and the data were harmonized. And you can see here in the table an overview about the different cohorts. And this MORGAM project is the basis for the BiomarCaRE project, which is an EU FP7 collaborative consortium, which is the biomarkers for cardiovascular risk assessment across Europe. So we already had the MORGAM database with harmonized or mainly harmonized the clinical data and phenotypic data, and BiomarCaRE, as the title says, it's the biomarker study on top of these clinical data. We also enlarged the cohorts that were used, and here in this graph you can see the different cohorts that were gathered in or are gathered in BiomarCaRE. All the blue ones are population-based cohort. The red ones are disease cohorts, which we added within BiomarCaRE, and we also had some clinical trial. And you can imagine that based on this data and also biomarker assessment, that we can span from health to really disease and even treatment, and to see what could be the value of biomarkers within cardiovascular disease. The entire BiomarCaRE project was set up in 3 different molecules. In the first molecule, we wanted to measure and analyze in the different biosamples from the cohorts omics parameters. So that was mainly the metabolomics part. We had microRNAs measured, and we had a lot of different proteins measured. The project also included small and medium-sized enterprises, and here biocrates was one of our main partners. And via this, the metabolomics part was very, let's say, successful. And we then wanted to conduct a phase 1 assessment, let's say half of the cohorts, where we measured all of the different markers and omics parameters. Then we developed and validated statistical methods for risk assessment, and the biomarkers that were successful in the first phase went to a second phase in order to validate. And in the end, it was the aim to come up with a so-called BiomarCaRE panel of markers that might improve the risk prediction across Europe. So here again, it's an overview about the population-based, the disease cohorts and the clinical trials. And you see that we had from small size, let's say, cohorts to really large size cohorts, and which one we used in phase 1 and phase 2. This is an overview because it's important, or it was important for our BiomarCaRE project, about the follow-up of some of these studies. And you can see, for instance, Glostrup, or here this Scottish heart health study, that they were established in former times, so more than 40 years ago, and we had a very, very long-term follow-up. And the idea of the population-based cohort was that we have biosamples at the time point when the subjects still were healthy, and they were followed up for incident events, so newly developed cardiovascular events. And via the baseline sample when the subjects still were healthy, and we did the measurements of our biomarkers, we wanted to establish or assess the value of the biomarkers for the risk prediction. And that's why it was very, very nice that we have this long-term follow-up where you get a lot of events. And one, let's say, speciality of the BiomarCaRE cohort is that you saw that over 300,000 subjects were or totaling the BiomarCaRE cohorts. But to measure in 300,000 subjects metabolomics, that might not be very good feasible. And that's why within BiomarCaRE, we initiated calculated case cohort setting. And case cohort is not case control, but you use a cohort design, where you, let's say, gather all the different cases that you would like to have, and you add, let's say, kind of a control, but it's not a control, it represents the cohort itself. So you have this subcohort of cases, which are very much dense, and you have a subset of the full cohort, which represents then this case cohort set. And by that, we could condense or reduce the sample size, but still keep all the information of that entire cohort design. And by that, from 300,000 subjects, we in the end could use all information, but only had to measure 11,400 subjects. And that is important because in that cohort, we have measured also the metabolites. So this gives you an overview what kind of biomarkers we performed. So in all 300,000, we measured existing biomarkers that are relevant or at that time were somehow interesting for cardiovascular disease. We also had mainly from the SMEs within the project emerging biomarkers. That was on one hand a 4-plex proteomics marker from one of our partners. MicroRNAs at that time were emerging biomarkers and exosomes. And within the case cohort, we performed also emerging biomarkers, the biocrates metabolite P180 panel, and as I said, several microRNAs. So you can imagine that it was a lot of work to perform to measure the metabolites as well as all the other biomarkers. But it still is a large effort to perform the complex data harmonization, because each of the individual cohorts has a different, let's say, although they are population-based, let's say a slightly different design, and also how clinical data or phenotypic data are assessed. And it was a huge effort to do this data harmonization with different steps. So you had to assemble all the different information. You had to define variables, define also from a clinical point of view. And that also differed across Europe. So what is, for instance, stable angina pectoris? Is there a stable angina pectoris? And how to define blood pressure, for instance. And then we had one central database or partner who did the harmonization and also added everything to the database. And this is still ongoing for further clinical variables that are included. So this is somehow a process when you work with large scale cohorts that you need to take into account. It's not only to get the data and that's it, but there needs to be a harmonization step. So BiomarCaRE now, because it's now really a huge database with a lot of opportunities, offers on one hand a large resource of greatly harmonized data with a long-term follow-up. Biomarkers were measured centrally. That was done here in Hamburg. So we not only performed the metabolomics, but also all the microRNA assessments. All the protein biomarkers were measured only here in the laboratories. There are still biomaterials available. And we, within the project, we have established an analysis pipeline. So every time when new biomarkers are measured, this goes into the database, and then, let's say, basic statistics is established as a pipeline. And just some successes, let's say. Not only focused on, let's say, only cardiovascular disease and the risk assessment, but also how, for instance, alcohol intake and total mortality are associated, or the roles of lifestyle and clinical risk factors. So there is a huge opportunity to perform all different kinds of analysis. And one let's say, very important part within our group here in Hamburg is that we have a focus on biomarkers for the acute coronary syndrome or the diagnosis of cardiovascular disease, and in particular myocardial infarction, which is mainly based, of course, on patients. But we also wanted to assess whether you might have already a higher levels of troponins, which is a heart-specific biomarker that can tell you whether you are at risk for a future myocardial infarction. And that was now possible within the BiomarCaRE project, because as you can see here, we measured in these 300,000 subjects troponin, and you can see in the fifth or quintiles that the hazard ratio to get cardiovascular mortality or cardiovascular disease increases the higher the troponin values are. And these troponin values are actually not really high levels as you would have when you have a myocardial infarction. These are still low, but although they are low, the ones that have slightly higher levels, they are at great risk. And that was something which we then always intended to publish, let's say, or to analyze from health to disease to really the people who have myocardial infarction. And we were able to establish kind of a compass for the clinic and also for let's say, the physicians to gather the information for people who might be at risk. And in the end, we were able to add these data to the SCORE project, the risk prediction algorithm, which are currently used also within Europe. And that was something quite nice because I never had imagined at the beginning of the project that just, let's say, by measuring and having all these data that we would be able to have something which is really applicable across Europe. So now I would like to give you some examples of what we did with our metabolomics analysis. And this will be separated into metabolomics and the incidence of coronary heart disease, metabolomics and atrial fibrillation. And then I briefly will also talk about an EU project, a different one than BiomarCaRE, where we also have measured metabolites. So as I said, we are interested in cardiovascular risk assessment, and now the disease I will focus on is in general coronary heart disease. And here, similar to entire cardiovascular diseases, it's the risk assessment is based on classical risk factors, but it's quite heterogeneous and there must be something more, and there are still factors not known. And by that, we wanted to identify novel biomarkers and have measured with the help of biocrates the metabolites. And as I said, biocrates was a partner within our EU project, and that was a very nice partnership because it was not only to provide the assays, but there were really visits back and forth to Innsbruck or from Innsbruck to Hamburg, and we had a very tight, let's say, partnership, and that also began already in which kind of instrument to use in order to perform the measurements. And I think that was only or part of the success was also because it was this tight partnership. So the aim of this study here with the coronary heart disease was to study the association between circulating metabolites and the incidence of coronary heart disease, and to see whether the combination of let's say normal biomarkers or classical risk factors to disease-associated metabolite would give us more information across our BiomarCaRE populations. And now this case cohort comes into play because we use this case cohort, and the analysis here was based on 10,700 subjects, so all with the incidence of coronary heart disease plus a subsample of this entire cohort. And we had over 2,000 incident cases of coronary heart disease. We performed the measurements of the P180 biocrates kit with 144 metabolites. and did the classical, let's say, Cox regression analysis and adjusted for the different classical risk factors, but also on the let's say size or distribution of the different cohorts that were included in the analysis. And these are the baseline characteristics, so you don't need to check into detail. It just gives you an overview. It was a normal population, so not very much diseased, normal age, and it was stratified according to 50-50, let's say, men and women. And we identified 4 phosphatidylcholines that were associated with the incidence of coronary heart disease. And then we went on to check whether these metabolites give us an information about the survival, whether it's high or low levels of these phosphatidylcholines would give you information on the coronary heart disease free probability. So the risk to get coronary heart disease. And as you can see, the higher the levels of these 4 phosphatidylcholines, the higher was also the risk across, I think it was 13 years to get coronary heart disease. And we also measured estimated the hazard ratios in a basic model and also the full adjustment model. And again, the low levels, you had a higher risk of to get coronary heart disease. So that was quite nice because there was an association of metabolites to the incidence of coronary heart disease, but a lot of biomarkers show these associations. And then we wanted to check what happens if we combine these markers into a panel. And again, we see when we combine all of them, of course, there is again the same information. Then we compared the individual and also this metabolite panel markers to the classical risk factors. And we wanted to see whether the strength of this association and the information behind this is similar or maybe even more than with the classical risk factors. And as you can see, so all these, even the individual phosphatidylcholines gave similar information than, for instance, the body mass index and the systolic blood pressure. So that was a nice information for us, and we further went on, and also wanted to derive a panel where we include the already established biomarkers with new biomarkers, for instance, troponin or anti-proBNP, but that panel did not add further information. So the metabolites, although they show an association to coronary heart disease and they have a similar strength than the classical risk factors, they did not really add more information towards the risk assessment than the currently established biomarkers. So we were able in this large population-based studies to evaluate the level of the metabolites, in particular phosphatidylcholines. And since this was this quite large cohort, we already had, let's say, validated the data. And it showed that we have a value for metabolomics for the improved risk assessment. But what to do else? These would be the next steps with that. Oh, it's sorry. I need to step in because there's some noise, let's say. So the next project we then went on was, and we wanted to evaluate the value of metabolites with atrial fibrillation. And maybe you can see it here. The normal heart rhythm, you have the, let's say, electrical conduction that the heart really pumps. This goes from here, from the atria, the Vorhöfe, across the walls into the ventricles. And then the heart regularly can pump. If you have atrial fibrillation, you have the problem that this electrical conduction is somehow gathered within the atria and it irregularly can go, or only irregularly goes to the ventricles, and then the heart cannot regularly pump, but it does like something like this. So, and atrial fibrillation is a common arrhythmia. It has a high comorbidity, morbidity, and thereby is a significant public health problem. And again, as with all cardiovascular diseases, some classical risk factors are already established risk factors for atrial fibrillation. But here in gray, you can see that there is still a large unknown part. And again, we wanted to see what the metabolites can add to this. We again used our BiomarCaRE case cohort set, where we had over 1,400 incident atrial fibrillation cases. And what we found was that acylcarnitine, the C18, showed a strong association to atrial fibrillation. And in that case here, we went on and we also validated our data in a second cohort, which was a disease-based cohort of atrial fibrillation patients from the Heart Otago from Australia cohort. It was a small cohort, but these were very much characterized and were patients with atrial fibrillation. And here we could validate this association of C18. And as shown here on the right side, so again, the higher the values, let's say, lower the risk, or no, the higher the risk of atrial fibrillation. So what are acylcarnitines? These are involved in the beta oxidation of fatty acids within the mitochondria. So they are needed for the energy production within the cells. And this C18, what we identified, it's a long chain acylcarnitine. And this is also an intermediate transport form for the fatty acid. So it somehow seems to play a role in the mitochondria. And it is also known that acylcarnitine can incorporate into the membrane of the cells. And our idea was maybe if there is too much, it incorporates in the membrane and thereby somehow, let's say, destroys the membrane. And we thought, okay, that might be quite nice. We not only identified this biomarker in association to atrial fibrillation, but we also want to see what happens on the molecular level. And thereby I initiated a doctoral thesis for, that was here, it's a girl, it's Julia Krause. And she performed then her doctoral thesis on this project, or the molecular evaluation of the effect of acylcarnitine on atrial fibrillation. And the model for which she used was an engineered heart tissue model together with our colleagues from the pharmacology. This is here Justus Stenzig. And what they did was they used really human cells and generated induced pluripotent stem cells and used these stem cells to then generate an, let's say, artificial heart model. You will see how it looks like. And this they did from the atria, cells from atria, but also cells from the ventricles. And then they put the C18 on top of these engineered heart tissue and checked different parameters which are related to the heart function. And I hope you can see it. So here on the left side, it's the untreated control, and this twitches like this. So this is a normal heart contraction. And when we add the C18 on that EHT, it just kind of flippers. And that was our first hint. I always like to show this because it looks like, let's say, atrial fibrillation. Of course, it's not, but the C18 does something to the engineered heart tissues. And so Julia then performed over the last years different assays, and she established a model where we think that the C18 can have a short-term, a medium-term, and a long-term effect. So short-term, in terms of engineered heart tissue, was a treatment of 20 minutes with the C18, and the C18 induced a positive inotrophic effect, which means that there was an increase in the heart force, let's say. So the heart could pump better. But if you treat it then for a medium-term effect, then these spontaneous contractions started. So which is shown here in the red bar, and also with this black line in the lower part. But this was specific for C18 because when we washed it out, when we just changed the medium, and then the spontaneous contraction were removed. So here, this was our first incident that there really was some arrhythmogenic potential. And when we treated the cells for a long-term effect, which we think might be accumulation of C18 over years, and then we found that the cells just die at their first start to do the spontaneous contraction, and then they die. So we developed now our current model that there seems to be an acute effect of C18 on the heart cells, which is also an interference with the calcium handling, which is needed for the cells to work, let's say, normally. But if you have too much of the calcium, or it's somehow distributed, and then they get arrhythmogenic, and there is a chronic effect, which leads then to a toxic effect over time. And when we, so our hypothesis is that over the years in a human situation, that over years, there might become an accumulation of C18, and that leads to the cell death of cells, to the arrhythmogenic potential, and in total then might lead to atrial fibrillation. And at the moment, we still work on this project. We investigate the effect of C18 on a therapy for atrial fibrillation. This is an ablation therapy, and we measure C18 before and after the therapy. And we also want to investigate where does the C18 comes from. And we have some information that it might come or might be influenced, let's say, by diet and also, of course, the microbiome. We also developed an optogenetic model for the engineered heart tissue, where we try to mimic atrial fibrillation. So you can put the light on, then the atrial fibrillation starts, and if the light is off, and then there is no atrial fibrillation. And here we again went back and measured the secretome on protein and also on metabolome levels. And at the moment, we have the first data where we identified 9 metabolites that might be influenced by the metabolite C18. And we also going back and want to see what is the genetic risk of higher C18. And by that, we want to establish a model of high genetic and low genetic atrial fibrillation risk, take these subjects and generate engineered heart tissues or induced pluripotent stem cells from these subjects with a high and a low genetic risk, and then see what happens if we treat them with the C18. So it's let's say a huge project which derived from the metabolomic analysis we conducted within BiomarCaRE, and which now includes even further large-scale cohorts. So as I said, we have these different resources of our cohorts, and I now showed examples here from the population-based cohorts. I also want to show you briefly examples of what we did with the disease-based cohorts. And there was one EU project, which we call the PREMAT CAD and Match project. And here we wanted to identify a dynamic omics signature, which might identify people at risk for myocardial infarction, but at a very early phase, who not have myocardial infarction, but are at the point where the oxygen is reduced within the cells, so they have an ischemia, and whether then the treatment or an early treatment might be good, and the early identification of these subjects based on multi-omics analysis. And these ischemic patients, on one hand, from cohorts, but we also had a mice model were measured or were used to measure metabolites and also proteomics. And we currently have the data for our mice models, where we had different groups which mimic, or again, the state from healthy to ischemic to myocardial infarction to heart failure subjects. And we measured here metabolites as well as microRNAs, and we vice versa want to see whether they can use a circulatory biomarkers in heart tissue or in the serum of these subjects and mice. And we have identified 4 metabolites which might be of relevance. So that just briefly should show you that you are not only, or that you can combine human cohort data also with some experimental models. Yeah. And by that, I'm already at the end. I think I showed you some examples of my, let's say, translational circle. But in the end, to unlock all these insights, there are, of course, a lot of challenges. And so let's say just briefly what I found is most relevant. So on one hand, this interdisciplinarity, which is quite nice, but you also need to be aware that the people of the different disciplines, statistics, the bioinformatics, the lab people, the biologists, and so on, the clinicians, they all talk slightly different languages. And you need to be aware of that you really need to put a lot of effort into that to understand each other. The biological annotation and interpretation of, in particular, metabolites, so far, in my opinion, is not so easy. We also had one study where we measured genomics, transcriptomics, metabolomics, and proteomics, and we wanted to, let's say, combine all of these data. But with the model we used, we were not able to integrate the metabolites into this model because there was not a good annotation which comes then from the transcripts or the genome to the metabolites, which of course, because they are metabolites, is not so easy. But this is something you also need to be aware of. Yeah, we had a lot of, let's say, costs for this entire project, but we greatly established our resource and were very, very successful with our industrial partnerships. We, as I said, within BiomarCaRE, we had a central biomarker measurements. So all the different samples were sent to our lab. So that was also a huge effort. But in the end, I would think this central measurement is way better because you have it in one hand. And if there are mistakes done, then it's always in one laboratory and not at different places. Yeah, and in the end, I somehow think that I showed you the different approaches we took, and we have some results, but how now to translate it into the clinical practice? So what is the added value of these biomarkers? This is something that still needs to be established. Yeah, and these are some pictures. This here is the BiomarCaRE group where we had our annual meeting in Venice, and somewhere there is also Manuel Kratzke from biocrates who was always participating here. It was very nice. And you can also see that for this large-scale project, there are a lot of people and different cohorts involved, and this is something which was very, very nice. But you also have to keep everybody, under one head, let's say. And by that, I really thank you for the opportunity to present our data, and looking forward for some questions.